All right, chapter 13 is all about meiosis. And in a more general sense, what we're going to be talking about here is the alternation of generations in the life cycle of eukaryotic organisms, or multicellular eukaryotic organisms. So what I mean by life cycle, before we jump into this, what I mean by life cycle is the series of stages that a multicellular eukaryotic organism goes through, for, let's say, for humans or for animals, something you're familiar with, right? There's the full-grown organism, right? That's me. And that organism will make basically single cellular organisms, right? In my case, sperm, which could unite with the egg from a female to make another cell called a zygote, and then that zygote will divide and grow up into another human. And so that's what I mean by life cycle. And plants do something very similar, and fungi do something kind of similar. But before we get into that, you need to understand what's going on with the chromosomes inside a cell. So, one thing we've talked about, but we haven't really fleshed out, is the idea of homologous chromosomes. I talked about this when we were talking about mitosis in the last chapter. Another name for these, I'll shorten them. Right? It's a lot easier to just say homologs. So if I say homologs, I mean homologous chromosomes. So what exactly are they? Well, they come in pairs. You know what a chromosome is, right? Because you watched and paid attention to the chapter 12 lecture. You know what a chromosome is. And you know that chromosomes can come in pairs. Homologous chromosomes are the same length. They have the same centromere location. They have the same gene location. So they have the same genes, homologous chromosomes do. They have the same genes in the same location. But, and this is important, they are not identical. And you may be looking at this thinking, well, if they've got this, they're the same length, they got the same centromere location, they got the same genes, how are they not identical? You'll see what I'm talking about, and I think you may intuitively already understand this. For any gene, you can have, any different number of versions of that gene. So one chromosome may have one version of the gene, and another chromosome may have the same gene but a different version of it. And we'll go into what those are in a little bit. So these homologous chromosomes can be visualized. So if you look at the first slide on the chapter 13 PowerPoint, what you're looking at there is, we call this a karyotype. And it's just a visualization of all the chromosome pairs in a eukaryotic cell. And so you can visualize the number of chromosomes, and you can see how they pair up. So this particular karyotype that you're looking at is of a human female cell. And so you can see that there are 23 pairs of chromosomes. Humans Humans have 46 chromosomes, usually. There are exceptions. We're not going to talk about the exceptions. So humans have 46 chromosomes. 44 of them are what we call autosomes. Autosomes are just non-sex determining chromosomes. So you've got 44 autosomes and two sex chromosomes. So really, you've only got 23 unique pairs of chromosomes. And so that's why when you're looking at that, that karyotype, you can see there's that first pair, that's number 1, and then 2, 3, 4, 5, 6, and so on, up to 22, and then the 23rd pair of chromosomes is, you know, either XX or XY, usually. So yeah, humans got 46 chromosomes with 23 pairs, 44 autosomes, 2 sex chromosomes. This particular cell, this particular karyotype, is of a diploid cell. Diploid just means 2 sets of chromosomes. So one thing that I don't want you to get confused is this difference between pairs of chromosomes and sets of chromosomes, right? When I talk about a pair of chromosomes, I'm talking about a pair of homologous chromosomes. When I talk about a set of chromosomes, I'm talking about, in humans at least, I'm talking about 23 chromosomes, right? 1 from each pair. So if a cell has 2 of each pair, or 2 of each set, then it's a diploid cell. If you only have 1 set of chromosomes, then you're looking at a haploid cell, which only has 1 set of chromosomes. So haploid cells don't have chromosome pairs, right? A haploid human cell only has 1 chromosome number 1, 1 chromosome number 2, 1 chromosome number 3. A diploid human cell has 2 of each. So if you're looking at a haploid karyotype of a human cell, then you would only see 1 of each of those pairs, right? We abbreviate. We have a good abbreviation for all these. Diploid, we abbreviate 2N. And then haploid, we abbreviate N or 1N. And N is just the number of chromosome pairs. So for humans, N is 23. So a diploid human cell has 46 chromosomes, right? So the N and the 2N really just refer to the number of chromosomes in that cell, where N is the number of chromosome pairs. Make sense? I don't know why I said that. Yeah, it makes sense. Okay. So, this chapter really just is, the point of the chapter is to clarify how different types of organisms reproduce. And when you reproduce, you're basically passing on those chromosomes to the next generation. And very broadly, there's only two strategies to do it. There's the simplest one, asexual reproduction. All right. In asexual reproduction, the offspring receives one full set of chromosomes from a single parent, right? So, a lot of, if you were to take a clipping of a plant, right, and then just plant that in the ground and it grows up into another plant, right, that's kind of like asexual reproduction, because it's really just you're making a clone of the original organism. So in asexual reproduction, the offspring receive all their chromosomes from a single parent. All right, that's easy to conceptualize because you know how mitosis works, right? It's just, you start with one cell and then it makes a copy of its DNA, and then those two copies split off into two daughter cells, and that's asexual reproduction. But sexual reproduction is a little bit more complicated, because sexual reproduction, in sexual reproduction, the offspring receive one set of chromosomes from each parent. So in humans, right, you received one set of chromosomes from your father and then another set of chromosomes from your mother. And those two sets were combined into one cell. So in order for this to happen, your mother, your mother's ovaries basically took a diploid cell and divided it up into multiple haploid cells. And then the same thing happened in your father's testes, and then those one haploid cell from your mother and one haploid cell from your father fused together to make a diploid cell called a zygote, and then that cell divided up to become you. So it's a little bit more complicated, and we'll go into the details of that in a little bit. But for right now, we'll just say the offspring of sexual reproduction receive one set from each of the parents. And we'll take it one step further. We'll say that the offspring of sexual reproduction arise from the fusion of two haploid cells. All right, fusion of two haploid cells to make a diploid cell. So, for sexual reproduction, there needs to be a way to take a diploid cell and divide it up into haploid cells, right? Most of the cells of your body are diploid, but if you're going to sexually reproduce, then your body has to make haploid cells. So the process by which that happens is called meiosis. So in meiosis, you're starting with a diploid cell, and then the diploid cell will replicate its DNA, and then that diploid cell will go through meiosis to create 4 different haploid cells. So very broadly, it looks like this. All right, here's our starting diploid cell. And in this diploid cell, we use different colors. For the sake of simplicity, we're going to say this diploid cell only has one pair of homologous chromosomes. And remember, I said that homologous chromosomes are not identical, right? That's why I'm using different colors here so you can see what happens, right? This chromosome is not identical to this chromosome. They're homologous, but they may have different copies of each gene. So I'm going to make them different colors. So there's the red one and there's the black one. But these are both homologs. All right? Okay. So this diploid cell is going to go through meiosis, but right now it's in G1 phase. Before we can do meiosis, this cell has to go through S phase, right? And if you remember, what happens in S phase is all the DNA gets copied. So after this cell goes through S phase, now it's got 2 copies of each chromosome. All right. So goes through S phase, replicates all of the DNA. Now it's got 2 copies of each homolog. And then the process, we're not into meiosis yet, right? The first step of meiosis divides up the homologs. The homologs will separate. So we call this step meiosis 1, right? There's 2 steps to meiosis. There's meiosis 1 and meiosis 2. In meiosis 1, the sister chromatids stay together. So remember, these are sister chromatids, these are sister chromatids. These are homologs. Understand the difference. So during meiosis 1, the sister chromatids stay together, but the homologs separate. So this red pair of sister chromatids will go into one cell, and stay together. And then the black sister chromatids will go into another cell and stay together. All right? That's meiosis 1. The sister chromatids stay together during meiosis 1. And then each of these cells will go through meiosis 2. All right. In meiosis 2, now the sister chromatids separate, and we get another round of cell division. So now these 2 chromatids will separate, and then these 2 chromatids will separate. So in meiosis 2 is where the sister chromatids actually break apart. One thing that you can't do here, is you can't mix up. Don't think that meiosis is just mitosis with another round of the cell division. That's a temptation. It's a mistake that a lot of students make, is they look at this and they think, okay, well, this is just mitosis, and then there's just another round. That's not the case, right? In mitosis, the sister chromatids break apart, right? There's only one round of division in mitosis, and in that round of division, the sister chromatids will break apart. In meiosis, the sister chromatids don't break apart until the second round of division. So this first round of division, I would say go ahead and do this. Hit pause and go look at your notes from the last chapter and compare meiosis to mitosis, and you'll see how it's subtly different. It's not just mitosis with another round of division. So hit pause and go do that and come back. All right, you back? You with us? All right, good. So we look at the phases of meiosis. The meiosis phases are very similar to mitosis. And in fact, they have the same names, but they're just named, instead of prophase, metaphase, anaphase, telophase, they're named prophase 1, metaphase 1, anaphase 1, and then prophase 2, metaphase 2, etc., etc. So there's prophase 1, metaphase 1, anaphase 1, telophase 1, and then prophase 2, metaphase 2, anaphase 2, and telophase 2. Right? So you go through prophase 1, 2, 3, 4, or prophase, metaphase, anaphase, telophase, and then prophase 2, metaphase 2, anaphase 2, telophase 2. So this all happens during meiosis 1, and this all happens during meiosis 2. But the phases aren't exactly, they have the same name as the phase in mitosis, but what's actually happening is a little bit different right from the start. All right, and we're going to start with prophase 1. All right, so in prophase 1, each of the homologs will just line up next to each other. So right off the bat, it's a little bit different from prophase and mitosis. So here's one homolog, right? These are sister chromatids. The whole thing is a homolog. And here is the other homolog. And in prophase, they line up right next to each other. All right, so in prophase 1, they line up next to each other, and then they do a thing called crossing over. And if you look at the next slide, then you can see what I'm talking about. What happens is that at one point along the length of this chromosome, this chromatid and this chromatid will cross over and then actually they'll get clipped right there where they cross over and then recombined with each other. So what you'll see is that they'll get clipped at one point, and then this chromosome will be recombined with that one, and then, or I should say this chromatid, and then this chromatid will recombine with that one, right? This does two things. One is it allows these homologous chromosomes to stay stuck together during meiosis 1. Or, until the cohesin proteins are cut, right? So now the cohesin proteins that were holding together these two chromatids, they're also holding together, up here, they're holding together the homologous pair, right? Because of that crossing over event, now both of these structures are held together. That's one thing that crossing over does. And the other thing that crossing over does, and we'll talk about the implications of this later on in the chapter, is if you look, what it did is it took the end of this chromatid and stuck it on the end of this chromatid. So if you look, we've got the red one, red, 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 and then the crossing over event, and then blue. So it kind of shuffles the deck, right? It rearranges the genes on this homologous pair. So this is why when I was talking about homologous chromosomes, I said they're not identical. This is why, right? Because there are all these crossing over events. You can look at the picture on the PowerPoint. You can see the crossing over happened for this particular pair 1, 2, 3, 4, 5 different times, right, along the entire length. I've only drawn one crossing over event here, but it can happen, you know, as you can see in the picture, 5 times. And it's totally random, so it can do a lot of, it can go a long way towards shuffling the deck genetically. But yeah, that's the big thing that happens in prophase 1 is crossing over. And this is the result of that. So now the homologs are held together by cohesin. So in metaphase 1, all right, we're going to zoom out on that cell. We still only got, what were we, red and blue? All right, there's our crossed over homologs. Right, in metaphase 1, all the chromosomes align at the metaphase plate. Very similar to mitosis. But then in anaphase 1, the homologous chromosomes separate. So now make sure I get this the same. All right. I could have drawn them farther apart, but whatever. All right. In anaphase, they separate. And then in telophase, they'll split apart into two daughter cells, right? One has one of the homologs, and then the other one has, well, dadgummit, I did it wrong. There we go. All right. So that's telophase 1. So this is that first round of division, right? Metaphase, anaphase, telophase 1. All right. And then in the second round of division, you've got metaphase 2. Again, the chromosomes will align at the metaphase plate. I'm not going to draw our crossover chromosomes anymore because that's tedious. You already know that happened. So in metaphase 1, again, they all align at the center of the cell. And then in, or metaphase 2, they all align at the center of the cell again. Anaphase 2, in anaphase 2, now the sister chromatids are cut apart. Right? And then in telophase 2, this cell is split apart, and then this cell is split apart. So now you have 4 daughter cells. Each one with 1 copy of that chromosome. So each of these 4 cells is a haploid cell. They're not diploid anymore. So this is how, you start. Remember, we started with 1 cell that looked like this. Right? This was our diploid cell that we started with. And now we have 4 haploid cells. 2 of them have 1 copy of the chromosome, and 2 of them have the other copy of the chromosome. 2 of them have the blue copy, 2 of them have the red copy. Whereas here in our diploid cell, we had 1 blue and 1 red. All right. In many organisms, the product of meiosis is a type of cell called a gamete. Right? So these 4 cells that I wrote over here, right? They could be a cell called a gamete. And what a gamete is just it's a haploid cell that can combine with another haploid cell to make a new diploid cell. And when a gamete from a male combines with the gamete from a female, then what you get is a diploid cell called a zygote. So a zygote is just a diploid cell that is produced by the fusion of two gametes. Right? So in humans, your gametes are either sperm or egg cells. So a haploid sperm and a haploid egg fuse together to make a zygote, which is a diploid cell, and then that zygote can then divide many times to become a full-grown human. Right? So that's one way to do it. That's one possible outcome of meiosis. Another possibility is that meiosis doesn't produce gametes. Instead, it could produce a cell called a spore. All right? A spore is a haploid cell that can divide by mitosis to make a full-grown organism. Right? So a lot of plants can make spores, like most plants can. So the product, don't think that the product of meiosis is always a haploid cell that's going to combine with another haploid cell. Sometimes meiosis produces a haploid cell that can then just go through mitosis over and over again to make a multicellular individual. So not all mature eukaryotes are diploid. Not all mature eukaryotes are diploid. So I've kind of alluded to this, but different types of organisms have different types of life cycles, right? You're probably pretty familiar with the human life cycle. I think I touched on it earlier, right? So I'm a full-grown human male, so my testes can produce haploid cells called sperm. Those are my gametes, and then the sperm can fuse with the egg from a female to make a zygote, and the zygote is diploid, and so the zygote can divide and divide and divide and eventually grow up into another human, God forbid. That's an example of an animal life cycle. Right? So just to summarize all that, you start with gametes. You got a female gamete, which is haploid, and a male gamete, which is also haploid. They combine together in a process called fertilization to make a single diploid cell. So you get a set of chromosomes here and a set of chromosomes here. They're combined into one cell. Now you got a diploid cell called a zygote. And then that zygote can divide many times via mitosis to make a full-grown diploid organism. Here we are. Right? A full-grown diploid organism that's multicellular. And then at some point during this organism's life cycle, it will probably start to produce gametes. And it produces gametes via meiosis. I'm just going to abbreviate meiosis, M-E. All right, so it'll produce gametes via meiosis. Those gametes are haploid. And then this gamete can then go on to fuse with another gamete to make a zygote, and so on and so on. So this is a typical animal life cycle, right? And it's quite a bit simpler than a plant life cycle. But I want to use this to introduce this idea of alternation of generations. So think of these gametes as their own generation. Think of them as single cellular organisms that are human, or whatever species this animal is. So you have a haploid generation, and then two haploid cells of that generation will combine to form a zygote, which is a diploid, or at the beginning of the diploid generation. And so you have this alternation of generations between haploid, diploid, haploid, diploid, haploid, and diploid over and over and over again. And you'll see this again in the plants. For sexually reproducing organisms, you're always going to see this alternation of generations. And in animals, it's pretty simple. Plants are a little bit more interesting. It's going to start out the same with two gametes. So we've got a female gamete and a male gamete. And again, these gametes can fertilize to create a zygote. And that zygote will divide via mitosis to produce a multicellular diploid organism. So far, it's all the same. This multicellular diploid plant is called a sporophyte. All right? You go look out your window, look at a plant. First plant you see is probably a sporophyte, right? Trees, these are sporophytes. These are multicellular diploid organisms. Then that tree, actually these dotted lines are kind of cluttering things up, I think. I'll erase it. Okay. So that multicellular sporophyte can produce spores via meiosis. So here's where it gets different, right? The sporophyte will produce spores via meiosis, and then those spores, if you remember, a spore is a haploid cell that can go through mitosis to produce a mature individual. So that spore will then go through mitosis to produce another multicellular organism, but this time it's haploid. This one is called a gametophyte because it will produce gametes. And so here, this haploid organism is going to produce gametes not by meiosis, but by mitosis, right? And then the gamete will combine with another gamete to make a zygote. So in plants it's a little bit more interesting, right? So I'll recap because I realize it is kind of confusing. All right, so it starts out the same with a female gamete and a male gamete. They fertilize one another to make a diploid zygote, and the zygote will divide and multiply and grow up into a mature multicellular diploid organism, right? A tree, a shrub, or whatever kind of plant you like. And then that plant, some cells in that plant will go through meiosis to produce spores, and then the spores will grow up into a mature haploid individual called a gametophyte, right? Sporophytes are called sporophytes because they make spores. Gametophytes are called gametophytes because they make gametes. And gametophytes make gametes not by meiosis, but by mitosis because they're already haploid. And then those gametes can combine with another gamete. So again, you've got an alternation of generations here. It's a sexually reproducing individual, so it's going to go through phases of diploid and haploid, right? From here to here, there's your diploid generation. Diploid, haploid. Diploid, haploid. And so plants will go through these phases of diploid and haploid and diploid and haploid. And so you may be asking yourself, how come I never see these? And the reality is, most plants have actually evolved to the point where the multicellular haploid organism is buried within the flower of a plant. There are some exceptions. Ferns. Ferns are probably the best example to really observe all these generations, because what will happen is, ferns, if you look at the underside of a fern leaf, you can actually see the casing around the spores, so you can see the spores all the way down the stalk of that leaf. That spore will fall off the bottom of the leaf, right, and grow up into a multicellular gametophyte, and it's actually a separate plant, and you can see it, and they're pretty small, you know, but they'll grow up into these mature, very small plants that can make gametes, and then the gametes combine with another gamete, and that grows up into a mature sporophyte, which is what you actually see when you look at the fern. Right? When you buy a fern from Home Depot, you're buying the mature sporophyte. So yeah, plants, they also go through this alternation of generations, and they go through different phases of sexual reproduction and asexual reproduction as well. So the sporophyte will reproduce asexually, and then the gametophyte will reproduce sexually. I've got in my notes the life cycle for a fungus. I'm not even going to touch on that. Fungi life cycles are weird and complicated. And you'll probably get into it in 1108, but I'm not going to worry about fungus life cycles here. Okay, so one thing that all of these reproductive strategies have in common, right, whether it's plants or animals, is when you look at the phase of sexual reproduction, where you have a gamete from one organism and a gamete from another organism combining together to form a new organism, that's going to produce more genetic variation, right? So there's a number of reasons for that. There's a few different reasons for how that produces genetic variation. All right, sources of genetic variation in sexual reproduction. Number one, we'll call it independent assortment of chromosomes. In metaphase 1 and metaphase 2. Here's what I mean by that. Let's say there's a metaphase 2, right? So you have two sister chromatids, right? And they'll line up in the center of the cell during metaphase 2, right? They could line up like this, or they could line up like this. It's totally random, right? Whether they line up like this or whether they line up like this. So which chromatid goes to which daughter cell is completely random. All right, so let's look at how this actually introduces genetic variation. For example, let's look at a cell that has two pairs of homologous chromosomes. So it has two pairs of homologs. Right? It's got a purple and green pair, right? These are homologs. They're not identical, that's why they're different colors. and then another pair of homologs that are red and black. And again, these homologs are not identical. That's why they're different colors. So, we're looking at a diploid cell here with two pairs of homologous chromosomes. And if this diploid cell were to go through meiosis to produce gametes, there are 4 possible combinations of chromosomes that can go into the gametes. Right? 1, 2, 3, 4. All right? You could get a green one and a red one, right? Because the gamete is going to get one from here and one from here. So we could get green and red, or we could get green and blue. Or you could get purple and red, or purple and blue. All right, so those are all of the different combinations that you could make if you were making gametes from this cell, right? So these are all the possible haploid gametes. So each of these would make a genetically unique offspring, right? So if you have only 2 pairs of homologous chromosomes, there are 4 possible gametes. So the number of possible gametes depends on the number of homologous chromosomes. And it's just a simple calculation. It's 2 to the power of n. So in this case, n is 2. There are only 2 pairs of chromosomes. So it's 2 squared. 2 squared equals 4. There are 4 possible gametes. Right? If you remember, humans don't just have 2 pairs of chromosomes. Humans have 23 pairs of chromosomes. So the number of possible combinations of chromosomes that you can make in the gametes produced by your body is 2 to the power of 23. If you punch that into a calculator, it's 8,388,608. You don't need to know that number. I'm just trying to illustrate for you all the different combinations that can happen, right? There's almost 8.4 million different combinations of chromosomes that can go into each one of your gametes. But that's not even the end of the potential variation here, right? Because if you mate with another individual, right, yeah, you have almost 8.4 million different combinations of chromosomes that can go into your gametes, but so does the individual that you're mating with. So the chances of getting the same combination twice, if 2 individuals mate twice, getting the same combinations in each of those siblings is actually 1 in 1 over 2 to the power of 46, right? Because you're actually looking at 2 sets here. It's not 1 to the power, or 2 to the power of 23, it's 2 to the power of 46. So that comes out to 1.42 times 10 to the negative 14th, or 1 in 70 trillion, right? That's the chances, 1 in 70 trillion, right? That's the chances of getting the same combination of chromosomes twice from 2 individuals mating twice. But that individual would still not be identical to you, right? Let's say if you're an only child and your parents decide to have another kid and they have one more kid, right? That sibling of yours has a 1 in 70 trillion chance of getting the same combination of chromosomes that you got. But that individual is still not genetically identical to you because of the second source of genetic variation, which is crossing over. Even if they got the same combination of chromosomes as you, those chromosomes wouldn't be identical because crossing over is random. And remember, crossing over rearranges the organization of genes that are on the homologous chromosome. By the way, I don't need you to remember this number, right? I know I say if I write it on the board, I want you to know it. I was just trying to illustrate how much variation, how powerful that independent assortment is in creating variation. Right? So, the second source of genetic variation in sexual reproduction is crossing over during meiosis. And what that does is it produces recombinant protein, or recombinant chromosomes. That have maternal and paternal genes. So these are 2 words that I haven't used yet. You could probably figure out what they mean if you think about it. Maternal just means you got that chromosome from your mother. Paternal just means you got that chromosome from your father. So in each one of your cells, you have 2 sets of chromosomes. You have one set that you got from your father, and then another set that you got from your mother. So we'll do it, I guess. So we'll say maternal is purple, and then paternal is here in red. So during meiosis, when your maternal chromosome, right, it's been replicated. You know, my markers are dying today. All right? And your paternal chromosome, in red, right? They cross over. So what you end up with is your maternal chromosome with a length of paternal on it, and then your paternal chromosome with the length of the maternal chromosome on it, right? So what this does, it's, you know, like I said earlier, it's kind of like shuffling the deck between each generation. All right, so that's another very powerful way to introduce variation in the genetic pool between each generation of a sexually reproducing organism. And then the third way, we'll call it random fertilization. Basically, what it comes down to is that all gametes have the same chance at fertilization. All right, you're probably aware of this, but organisms don't just produce one gamete when they need to reproduce, right? Usually they make a whole bunch. You might notice, it's March 20th right now. You're probably not going to be watching this, let's be honest, until April, right? So it's April probably right now. And if you look out the window, you might notice on your car there's this thin layer of yellow, and it's just caked in yellow because you haven't been driving it for 2 weeks because you've been social distancing. And, so your car is covered in this yellow film. That yellow film is the male gametes from trees, right? And so what they do, it's a pretty interesting strategy for reproduction if you think about it. They just cover every surface near them in their gametes. And each of those gametes has an equal chance of finding its way to the flower where it can fuse with a female gamete. So, it's totally random which gametes actually go on to fertilize another gamete and produce an individual. So anyway, that's the end of chapter 13. Go wash your hands.